Hello and good afternoon friends, welcome once again to CEC-UGC lecture. Dear friends, in our previous lecture we discussed about antibodies, their structure, their formation, their types. Today in this lecture we are going to discuss about antibodies and antigens reaction, as well as we are going to discuss about the applications and immunoassays developed for diagnosing various diseases. And for this we have again with us Dr. Amit Bhattacharya. As you all know that he is assistant professor in Ramjas College, as well as I would like to tell you about him that he has carried a part of research at Nottingham University, as well as he applied many of these immunoassays in his research work. So first of all, I would like to welcome our guest, Dr. Amit Bhattacharya. Hello sir, welcome to the EduSet lecture. As we discussed a lot about antibodies yesterday, and today we are going to discuss about how antigens and antibodies interact. Yeah. So we basically wish to know through you that how they basically interact, as well as you told us about this assay kit also. Yeah. What is this? Please explain.
Yeah. So today's lecture I've fragmented into the principles of the antigen-antibody interactions, how exactly antibody and antigen interact, then subsequently by using these principles how the different immunoassays have been developed which are being routinely used for diagnosing the disease conditions or checking the antibody level in our body conditions. So before starting into that one, let's come to what exactly an antibody is. So antibody is a immunoglobulin which is made up of a protein. We discussed it yesterday. It has got two chains. One is called as a heavy chain, the other one is called as a light chain. The heavy and the light chain on the N-terminal region contains a region which is called as a variable region. So this is the region through which it binds to the antigen. And subsequently the other part of after the variable region is called as the constant region. So both these heavy and the light chains are interconnected to each other with the help of a disulfide linkages, and further both these heavy chains are further linked to each other with the help of a disulfide linkage which goes at the hinge region. Now what exactly an antigen is? So antigen can be anything, it can be any foreign particle or any protein which is present in our body. So an antigen on its surface bears many of the antigenic determinants which are called as the epitopes. So if you can see the pictures over here, there are three epitopes which are present. So these epitopes are critical in eliciting or evoking the immune response within our body. So what happens is whenever there is an antigen which gets into our body or foreign particles which get into our body, these antibodies get elicited or start getting synthesized from the plasma cells. So over here I have shown an antigen molecule which contains different surface epitopes present onto it, and each of these surface epitopes have got a different antibody which binds to them, and each of these antibody in the front or the N-terminal end contains a antigen binding sites. So with the help of these antigen binding sites, these binds to the epitopes. So the association between the antigen antibody involves various non-covalent interactions, and the major regions which are involved in this antibody are the N-terminal variable regions, and while in the case of the antigens the epitopes are involved. So these reactions are basically dependent on the non-covalent interactions and depends upon a number of other factors. So what happens is whenever in our body there is a antigen or a bacteria gets into our body, it's go through the circulatory system and through the lymphatic or the blood vessels, it is transferred to the B cells. It elicit or invoke the B cells which subsequently goes on to form the plasma cells. The plasma cells, what it does is it immediately starts secreting the antibodies against these particular antigens, and what happens is these antibodies goes and attaches to the epitopes which are present on the surface of these bacterial cells or the antigen. So they immediately form a mesh-like or lattice-like thing which precipitate out from the body reaction. Now based on these particular principles and the specificity of the antigen-antibody interaction, there have been development of a number of immunologic assays. These immunologic assays are routinely used for diagnosing diseases such as cancer, HIV, malaria, dengue, chikungunya, as well as it helps us to detect and identify which particular biomolecules are present in our body at an abnormal condition. So all these assays are based on the basic principle of antigen-antibody interaction. Before going into the antigen-antibody interaction or the various immunoassay, let's come into what exactly antigen-antibody interaction is all about. So the antigen-antibody interaction is basically based on the interaction which is non-covalent. So there are different non-covalent interactions of the bonds which are formed between an antigen and antibody, which are basically hydrogen bond, ionic bond, hydrophobic interactions, and van der Waals interactions. And subsequently what happens is these interactions are weak, so an antibody is called as a high affinity antibody if the interaction patterns between these antigen and antibody it's much more greater than the other patterns, and the distance on which these antigen antibody interaction takes place is about 1 angstrom unit. Now, these are the various forces which plays a major role in the interaction of the antigen and the antibody, which are hydrogen bond, where there is a sharing of a hydrogen bond between the electronegative oxygens, then there is an ionic bond, then there are hydrophobic interaction as well as van der Waals interactions which are taking place. So all these forces are playing a major role in the interaction pattern of the antibody and the antigen reaction. Now, the antigen-antibody interactions are majorly defined on two major principles. One is called as the affinity of the antibody, the other one is called as the avidity. So the first phenomenon which came in was called as the affinity of the antibody. So the affinity of the antibody is the combined strength of all the non-covalent interactions which are between a single antigen binding site present on the antibody and a single epitope present on the antigen. So if we see the picture over here, there is a single antibody and a single antigen epitope which are combining to each other. So that is called as the affinity of the antibody. Now, this affinity was not able to completely define or measure the entire capacity of an antibody, because the antibody-antigen interaction cannot be only defined to a single antigen binding site or a single epitope binding site. So there are multiple interaction patterns which happens over here. So the strength of the multiple interactions between the multivalent antibody and a multivalent antigen is called as avidity. So the avidity is the interaction pattern which gives us a better understanding of the measure of the binding capacity within the biological system. So the avidity over here, if you can see the picture, there are number of antibodies which are present over here and there are number of antigens which are present over here. So the avidity of the antibody is giving us a better picture than the affinity. So the avidity is the one which is now routinely used to measure the antibody-antigen interactions in the normal senses. Now, the first antibody-antigen interaction patterns were followed in the precipitation reaction. So the precipitation reaction for the first time used the antigen-antibody interaction pattern. Now, precipitation is actually the antibody and the antigen interaction in the aqueous solution, which forms a lattice that eventually develops to form a visible precipitate. So there is a visible precipitate which is formed. There are a number of factors on which this precipitation reactions are based on. The first major factor is the antibody has to be bivalent or multivalent, which means that the polyclonal antibodies have to be present. Means the antibody against different epitopes has to be present in that particular serum. The second major factor on which the precipitation reaction is involved is the antigen must be bivalent or polyvalent, which means that the antigen should have on the surface of it a number of epitopes which are present on the surface of it. So if we see two pictures over here, one is called a monoclonal antibody in which the antibody is against a single epitope, so there is no formation of any lattice or visible precipitation which is occurring. While in the case of the polyclonal antibody, we can see there are different types of antibodies which are present, all of which are getting attached to the antigen or the epitopes of its interest, and it is forming a mesh-like thing which is giving a visible precipitation over here. So this basic principle of precipitation reaction was used over here. Now subsequently this precipitation reactions gave a curve which was called as a precipitation curve, which was plotted between the antibody precipitated and the antigen added. So this curve revealed three major zones in which this antibody-antigen interactions were divided. The first zone was called as the antibody excess zone, where the antibodies were in much more higher concentration than the antigen. So if we see over here the picture, the antibodies are in much more higher concentration than the antigen, so there is no visible precipitation formed over here. The second zone is called as the zone of equivalence, where the antigen and the antibody are present in an optimum or at a correct concentration. So there is a formation of a lattice or the precipitation over here due to multivalent and multivalent antigen interactions over here. The third zone is called as the antigen excess zone, because over here most of the antibodies have been utilized in the equivalent zone, so we are left with only few antibodies, so most of the antigens are present over here, so the antigen concentration increases over here and there is no formation of any clear precipitation over here. So all these phenomena or the principles were utilized in subsequent development of the immunoassays, which were used for diagnosing various diseases or checking up various concentration of proteins or hormonal levels in our body. So the precipitation curve was showing three major zones, of which the zone of equivalence was of importance, where the exact precipitation line was formed due to the optimum condition or the optimum ratio of the antibody and the antigen concentration. Now, this precipitation phenomenon was used in the precipitation reaction in the gel. So it yielded some basic phenomena or basic reactions which are called as the immunodiffusion assays. So these immunodiffusion assays gave birth to the first immunoassays which were able to determine the relative concentration of the antigens or the antibodies, and it was subsequently used to compare the antigens which are present over in the blood samples. It can also determine the purity of the antigen preparation. So by this assay there were two major assays which were developed. The first one was called as the radial immunodiffusion assay. The second one was called as the double immunodiffusion assay. So in the first one, which is called as the radial immunodiffusion assay, which is also named as Mancini, named after the scientist who discovered it, in which the immuno reaction was taking place in the agar, which are basically a semi-solid gel. So in the gel, the antibody was already incorporated. So the gel contains, or the agar contained, the antibody within it. And there was a well which was made at the center of this gel, into which the antigen or the test sample were loaded. Subsequently, what the scientists found is these antigens diffused out through these gels, and at a position where the concentration of the antigen and the antibody became the optimum or the correct, there was a formation of precipitin ring over there. So a precipitin ring was formed over here, and this prominent precipitin ring, if we can measure the area of this precipitin ring and compare it with the standard curve, we can tell the concentration of the antigen sample over here. So over here, if you can see, the test sample was simultaneously measured along with the standard sample. So the test sample was loaded in the well along with the standard sample, and if we can see, there are clear precipitin line formed against the standard and the test sample. And when this test sample is compared with the standard sample, we can easily make out that the test sample also contains a large amount of these antigens which are present in the standard 4 and standard 2 solutions also. Now, this assay is routinely used for checking the various abnormal conditions. For example, over here, the assay was used to check the concentration of beta-2 microglobulin in the serum. Beta-2 microglobulin is actually a small protein which is associated with the human leukocyte antigen, and it is present on the normal nucleated human cell. So it is present on the surface of the normal nucleated cell. And it has been found that these beta-2 microglobulin concentration increases in the serum in the various abnormal conditions of the disorders, which includes renal infections, acquired immunodeficiency syndromes, autoimmune diseases, transplant rejections, and subsequently it was found that they were also high in the leukemia condition. So If the patient samples are taken, it is loaded onto the well, and subsequently if the precipitin line is formed which can be compared with the standard curve, we can easily make out whether the patient is synthesizing a high level of these beta-2 microglobulin. If the patient is secreting a high concentration of this particular protein, which is easily visible to the precipitin line which is shown in the picture, so we can easily make out that the patient is suffering from one of these disease condition or the disorders. So the patient can go into the next level of the screening, what exactly the disease is and how exactly the diagnostics of the disease can start immediately. The next immunodiffusion method which was developed through the principle of precipitation was the double immunodeficiency diffusion method, which was called as Ochterlony method. This Ochterlony method, instead of using the antibodies within the gel, it was using the antibody which was also loaded along with the antigen in two separate wells. So in a gel, two separate wells were created, in which in the one well the antigen was loaded, in the other well the antibody was loaded. So in the agar medium, this antibody and antigen diffuse towards each other. So at a precipitin line where the condition or the zone of equivalence, where the concentration of the antigen or the antibody was optimum, there was a formation of a precipitin line. So by this particular method, we can easily make out what are the antigens which are present in the body, or what are the serums or the protein serums or the hormones which are abnormally produced in our body. So over here I have shown a picture how exactly this assay is done about. So first a agar plate is made. Into this agar plate different wells are made with the help of the troughs. So into these wells the antigens are loaded. So the antigen is basically loaded on the peripheral wells, while at the center well the antibody is loaded. There are different precipitin lines which are formed by this reaction. So there are different precipitation pattern on the outer Lony plate which can be found. Over here I have shown three major patterns which are found. The first one is the identity precipitation pattern, the second one is called as the non-identity, third one is called as the partial identity precipitation line. The first one is called as the identity because the two antigens which are loaded in the two wells are similar, and the antibody which is loaded in the well is the anti-1 antibody. So this anti-1 antibody is against the antigen which is loaded in both the wells. So both the wells are having the similar antigen, so there is a V kind of precipitin line which is formed. So we can easily say that both the antigens are same. If the antigens differ from each other which are loaded into the two individual wells, we immediately get a cross line which shows a X kind of situation. So over here in one of the well there is an antigen which is 1, and the other well there is a 2 and 4 antigen, and the antibodies against antigen 1, 2 and 4 are loaded in the antibody well. So after some time we can easily see in the gel there is a picture which is showing a X kind of picture, which is immediately showing that the precipitation pattern is a non-identity, and we can easily make out that this antigen in well 1 and the antigen in the well 2 are both different. The third precipitation line which is formed is called as the partial identity, in which both the antigens have got a overlap epitopes present to it. So the antigen 1 and the antigen 2 also has got a overlapping antigen 1 epitope present to it. So there is a partial identity between both the antigens, and over here what we get is a spur kind of precipitin line, and we can easily make out that there is some kind of identity between the precipitation found in the antigen well 1 and the antigen well 2. So all these methods gave about the precipitation reaction in which we can easily make out what are the antigens which are present in the blood serum conditions, or what are the antibodies which are present. The next major assay which was developed was called as the immunoelectrophoresis. This immunoelectrophoresis basically combined the electrophoresis and the double immunodeficiency diffusion method. So this immunoelectrophoresis basically used the electrophoresis first, in which the antigen sample which contained a number of antigens within that particular sample, they were first electrophoresed depending upon their charges. So the antigens, which are the proteins, can be of different charges. So they can be positively charged, they can be negatively charged, or they can be neutral also. So first these antigens are separated on the basis of their charge on a gel. So the first thing what happens is the electrophoresis which occurs. After the electrophoresis is complete, on the top of these gel, there is a trough which is created. So on this trough, there is the loading of the antibody which takes place. So the antibodies against these particular antigens are loaded onto this well, and subsequently these antibody and these antigens, as they diffuse from their position, there is a precipitin line which is formed, just like the same thing which happens in the double immunodiffusion method. So the double immunodiffusion method and the electrophoresis method were combined over here to develop the immunoelectrophoresis method. So over here, if we see, the electrophoresis is done, in which the antigens are loaded onto the well. Into this well, when the electrophoresis starts, they go towards the opposite pole, like the negatively charged antigen or the protein moves towards the positively charged, while the positively charged antigen or the protein moves towards the negatively charged. So they are separated on the basis of the charge, and subsequently what happens is there is a trough which is created on the top of the gel, and onto this trough the antiserum is loaded. So the antiserum over here is shown in blue in color. So when the antiserum is loaded, it starts diffusing through the gel towards the antigen, and the antigen also start diffusing towards the antibody. So at a proper condition, there is a formation of the precipitin line, and we can easily make out with the help of this precipitin line that these particular antigens or the proteins are present in this test sample. This electrophoresis are routinely used for checking the cancer samples. Over here I have shown a cancer sample in which the patient is suffering from myeloma cancer. Myeloma cancer is actually a cancer of the myeloma cells, in which there is a large amount production of the monoclonal IgG antibodies, especially the lambda light chain bearing antibodies. So the IgG antibodies, which contains the light chain lambda chains, are produced at a high concentration in the patients who are suffering from the myeloma cancer. So just to identify whether the patient is suffering from the myeloma cancer or not, what we can do is the patient samples are taken from the blood serum. The serum sample is identified from that blood thing, and then this serum is loaded onto the gel antigen well. Subsequently, these antigen well are electrophoresed so that the antigens which are present in the blood serum get segregated based on their charge. So the IgG and the lambda bearing chain or the IgM get separated on the agar gel. What happens is, after the electrophoresis is complete, on the top of the agar plate there is the loading of the antibody which is specific against, in the first plate, which is specific against the IgG molecule. Then the loading of the antibody takes place in the second plate, which is specific against the IgM antibody, and in the third plate the antibodies are loaded which are specific against the lambda light chain. So after the immunodiffusion diffusion is over, we can clearly see a precipitin line which is formed. So in the case of the myeloma cancerous patient, we can easily see that in the IgG there is a precipitin line which is formed, while in the case of the lambda light chain bearing precipitin, there is also a formation of the precipitin line, while in the case of IgM there is no precipitin line. So we can easily confirm that the patient is suffering from the melanoma cancer, because there is a high concentration of IgG and lambda light chain bearing precipitin line, which is showing that the blood serum are having a high concentration of these monoclonal antibodies present into them. So these immunoelectrophoresis and the immunodiffusion methods have been very useful in diagnosing the disease condition or the disorders which are present within our body. The next major reactions which are using this antibody-antigen interaction is called as the agglutination. So the agglutination reaction is basically similar to the precipitation reaction. In this, the interaction between the antibody and the antigen takes place, which results in visible clumping. One of the agglutination reaction which we routinely see is the blood group typing. So whenever we go to some pathological laboratory for checking our blood group, the medical representative does the agglutination reaction to check the blood grouping, what exactly our blood grouping is. So the blood groups were first identified by the scientist called as Karl Landsteiner in the year 1930. For his pioneer work in the year 1930, he was awarded the Nobel Prize for the discovery of the blood antigens which are present on the red blood cells. So this was the first time he discovered that what are the antigens which are present on the surface of the red blood cells, and for his Nobel work he was given the Nobel Prize for the discovery of human blood groups. So he discovered the human blood groups. Now all of us know that the human blood groups are basically of 4 types. So there are 4 blood group types which are present. The first one is called as type A blood group, the second one is called as type B, the third one is called as type AB, and the fourth one is called as a type O blood group. Now what exactly these blood groups are? These blood groups are basically dependent upon the antigens or the antibodies which are present in the plasma serum. So on our red blood cell, if there is a antigen A which is present, and in our plasma if there is a anti-B antibody which is present, then we say that the person is having a type A blood group. If a person is having a type B blood group, then on the surface of the red blood cells there will be a presence of antigen B, and in the plasma or the blood there will be a presence of anti-A antibody. If the person is of type AB blood group, then the person will having both antigen A and antigen B on the surface of the red blood cells, while there won't be any antibodies present in his plasma. While in the case of type O blood group, there won't be presence of any antigens on the surface of red blood cells. That's why there is neither A nor B antigens which are present, but the person will be having both antibodies against anti-A and anti-B. The type O blood group patients are many a times commonly called as the universal donors, because the red blood cells are having neither A or B antigens present to it. So as we take out the blood from the type O blood group donors and we transfuse them into the recipient, as there is no antigens which are present on the surface of the red blood cell, so there won't be any precipitation or agglutination reactions which will be taking place. While in the case of type AB blood group, if we see, there is a presence of antigen A as well as antigen B, but there is neither antibody A or neither antibody B which is present. So these type of blood group people which are type AB are many a times called as universal recipient, because they don't have any antibodies present in their plasma. So whatever blood we give to them, they won't show any kind of agglutination or rejection reactions, because there is absence of any antibodies which are specific to antigen A or antibody A. Now these particular blood grouping was used, the ideas were used to develop the blood group typing, in which we can identify what exactly a blood group is and what is the blood group of the patient is. Now before going into how exactly we do this blood grouping, in this what we do is, first we take out some blood from the fingers by needle pricking. Subsequently, there are different reagents which are available in the markets, which are anti-A antibody, anti-B antibody and anti-D antibody. So anti-A antibody are specific against antigen A, anti-B antibody are specific against antigen B, while anti-D antibodies are specific against Rh factors, which many a times we say that the person is having B positive blood group or a B negative blood group. So the positivity or the negativity is coming through the Rh factor. So if the Rh factor is present, it is called as a positive blood group. If the Rh factor is negative, it is called as a negative blood group. So there are 3 reagents which are present in the kit. One is is the anti-A antibody, the other one is anti-B antibody, and the third one is anti-D antibody. So there are three on slides. At three places the blood is dropped from the finger, and to each of them different reagents are added. So in the first one, the anti-A antibody is added, to the second anti-B antibody is added, and to the third anti-D antibody is added. So after mixing, what we can see is if there is a agglutination, visible clumping or agglutination is there or not. So if you see the picture over here, on the left hand side image, there is no visible agglutination over here, but on the right hand side picture, when we mix the blood and the reagent, there is a visible agglutination which is occurring, which shows that that particular antigen is present in that blood group of which the reagent is. So based on these agglutination or visible clumping, we can make out what particular blood group the person is of. So in the type AB, if there is an agglutination with both anti-A and anti-B antibody, which we can see in the picture, we can easily say that the person is type AB because he or she has got both antigen A and both antibody A on the surface of it. If the person is having type A blood group, then there will be agglutination or visible clumping only with the anti-serum against anti-A. There won't be any clumping in anti-B. So we can easily make out that the person is type A blood group. If the person is type B blood group, there will be a clumping or visible agglutination in the anti-B anti-serum, and there won't be any agglutination in anti-A anti-serum. So we can easily say that the person red blood cell contains only antigen B, not the antigen A. So the blood group is type B. And while the fourth one is type O, where there is no visible clumping or agglutination which can be observed both with anti-A or anti-B antibody. So we can easily say that the person is type O blood group. So by just looking at the agglutination or the visible clumping in very short span of time, we can easily check out that what exactly the blood group of this particular person is, and this hemagglutination or the agglutination reaction has been actively used in checking the blood typing. Now there is an anti-D which I told you, on the basis of this anti-D, the person is said to be positive or negative. So there are different blood groups which are there. The first one is a O positive, where there is a agglutination only in the anti-D well, so there is no visible clumping in anti-A, anti-B, so we can say that the person is O positive. Likewise, if there is no clumping in any of the well, we can easily say that the person is O negative. If there is only clumping in anti-A and clumping in anti-D, we can say that the person is A positive. Subsequently, we can say A negative if there is no clumping in the anti-D but clumping in the anti-A, and there are B positive, B negative. Likewise, there is AB positive, AB negative. So based on these things, we can easily make out what exactly the blood group of a person is by this particular kit. So this thing has revolutionized all the basic phenomena before going for any of the blood transfusion or the organ transplantation processes. Now, this basic agglutination in a reaction has been also used in the early types of the home pregnancy kits. So the home pregnancy kits which were discovered in the early period were based on this agglutination reaction, which contains the human chorionic gonadotropin hormone as well as the antibody against this HCG. What this kit basically contained is a latex bead containing HCG molecules to which the HCG molecules are attached, as well as there was an anti-HCG antibody. Now, a pregnant woman is said to produce a large amount of HCG in his body circulation. So these HCG are the one or the hormones which are checked or detected at the very early level to say that whether the woman is pregnant or not. So the first possible reaction if the woman is not pregnant is the urine of the woman is taken, it is put into the kit, into this kit there is a presence of anti-HCG antibody. So as there is no HCG hormones which are present, so there won't be interaction of antigen antibody. Subsequently, after the incubation process, the HCG carrier conjugate is added to it, and there is no visible clumping. There is a visible clumping which is formed in the case of non-pregnant women. So we can easily make out by the visible clumping that the woman is not pregnant, because there is no presence of HCG. While in the case of the positive reaction where the pregnancy has to be confirmed, is the urine sample is loaded onto the kit, as the urine sample of the pregnant woman will be containing the HCG hormones. So these HCG will immediately goes and binds to the antibodies which are present in the kit, the anti-HCG antibodies. We incubate it for some time, then subsequently when we add the HCG carrier conjugate, as most of the antibodies has been bound to the HCG which is present in the urine, so there won't be any visible clumping, and we can easily say that as the agglutination is inhibited or there is no visible clumping over here, we can say that the woman is pregnant. So this is one of the basic kits which were developed based on this agglutination reaction, but now most of the pregnancy checking kits which are available in the market are based on the ELISA, which we come into subsequent things. Now, one of the major disease which is coming is a dengue. So the dengue checking kit is also based on this immunochromatographic assay. In the dengue, actually the virus start developing within the body or the platelet cells, and these viruses actively synthesize or produce a molecule which is called as NS1, which means non-structural 1 protein. This non-structural protein 1 is a highly conserved glycoprotein, and it is essentially required by the dengue viruses, and it is produced both by the membrane associated as well as the secretory form of these viruses. So in the dengue virus infected patients, there is a high concentration of NS1 antigen which is present in the blood circulation. So if the scientists thought that if they can detect this NS1 antigen which is circulating in the dengue infected patients, they can easily check or confirm that whether the patient is suffering from dengue or not. So what they came up is a immunochromatography assay which was based on this antigen antibody interaction. What they did is, in a chromatographic strip, they loaded the antibody which was specific against the NS1 antigen which are produced in the dengue infected patients. So when the blood samples from these patients were taken and they were loaded onto the kit, and subsequently they were incubated onto this immunochromatographic plates, what they found is if the person is having the NS1 antigen, this antigen will combine with the antibody and give a clear precipitin line, or the precipitation or the agglutination will be formed and a line will be formed. So it will confirm that the patient is suffering from dengue. If the patient is not having any NS1 antigens within its blood circulation, there won't be any reactions which will be taking, and there won't be any precipitation line which should be formed. So if you see the picture over here, this is a single step dengue NS1 antigen test kit which is available in the market, which is based on the principle of the antigen antibody interaction. So over here, if you see the kit, onto the kit the blood sample from the patient is loaded, and subsequently after the loading of the blood sample, it is incubated for some time by addition of the buffer or the other dilutants which are available with the kit. And if the person is having the NS1 antigen present within its blood, there will be a antigen antibody interaction which will be taking place, and there will be a positive black line which will be shown over here, and in the case of the negative reaction there won't be any positive black line which will be shown. So just by looking at this simple test kit, we can easily make out that whether the person is suffering from dengue or not. So this kits, the single step, one step dengue NS1 antigen test kits are now readily being used to check most of the diseases like dengue. Subsequently, for the other diseases, the different types of test kits are also getting developed. Now, the first assay which was majorly developed based on these particular antigen-antibody interaction was called as a radioimmunoassay. Now this radioimmunoassay was discovered by two endocrinologists, Burson and Yellow. So they were first time they developed this assay which measured the hormone or the serum protein or the drugs or the vitamins concentration at a very low level. So for their innovative work, Yellow was awarded the Nobel Prize in 1977 for the discovery of the radioimmunoassay of peptide hormones. So what this assay is basically dependent upon to competitively binding of the radio-labeled antigen and the unlabeled antigen to the high affinity antibody. So these particular assay, the radioimmunoassay, basically detect and test the quantity of the antigens which are present in the test sample. So there is a radio-labeled antigen and a unlabeled antigen. So first what happens is in the well which are coated with the antibodies, the radio-labeled antigens are loaded. So these radio-labeled antigens goes and binds to all of these antibodies, and with a high affinity they bind to them. Now subsequently the test samples are loaded over here. So what happens is there is a competitive binding property of these unlabeled antigens to these antibodies. So what they do is they displaces these radio-labeled antigens from the bind side of the antibody and they get bound to these things. So there is a decrease of radioactivity which has been found. So this decrease in the amount of radio-labeled antigen bound to the specific antibody in the presence of test sample gives us a measure and determine the amount of antigens which are present in the test sample. So over here I have shown a picture in which the hepatitis B antigens are being measured. So just to quantify how much is the hepatitis B antigen, surface antigens which are present in the test samples, what happens is first there is a radio-labeled hepatitis B antigens which are coated onto these antibodies. So to all these antibodies the hepatitis B antigens bounds which are radio-labeled. Subsequently the test samples are loaded into these wells. As these test samples which are unlabeled, what they do is they competitively bind to these antibodies or the antigen binding sites, replacing the radioactively labeled hepatitis B antigen. So we can see that the hepatitis B, radioactively labeled hepatitis B antigens are replaced from here. So there is a decrease of the radioactivity over here. So with the help of a standard curve which is made through the bound percentage of the antibodies to the antigens and the concentration of the unlabeled hepatitis B antigen. So with the help of this standard curve we can easily extrapolate what exactly is the concentration of the hepatitis B antigens which are present in the infected serum, and through this one we can easily say that this much quantity of hepatitis B antigens are present or this much severity of the conditions are. Subsequently there were different assays which were developed based on these protocols. The first assay was based on which was called as the enzyme-linked immunosorbent assay. Instead of using any radio-labeled antigens, what this assay used, this assay used a secondary antibody to which an enzyme was attached. This enzyme was basically peroxidase or beta-galactosidase. So what this enzyme did is it converted the chromogenic substrate which was colorless into a colored product. So whenever we add this antibody and we add the substrate based on the antibody binding to the particular antigens, there will be a colored reactions which will be found. The first major type of this ELISA assay was the indirect ELISA. So the indirect ELISA is one of the major assays which are used to detect the presence of serum antibodies against the HIV virus in the AIDS infected patient. So in the AIDS infected patient, they are routinely checked how much is the quantity of the antibodies which are present in their blood circulation. So the blood serums are taken from the patients, they are coated or incubated onto the well which contains a antigen coated wells. Onto these wells, these antibodies goes and specifically binds to these antigen. Subsequent washes are done, and subsequently a secondary antibody which is a anti-idiotype of the primary antibody is loaded, which goes and binds to the primary antibody, and as the secondary antibody contains an enzyme which converts the colorless substrate, which is a chromogenic substrate, to a colored substrate. So there is a colored reaction which takes place, and the measurement of these colored reaction with the help of the specialized spectrophotometer help us to give us a measure that how much amount of these antibodies are present within the patient who is suffering from the AIDS or the HIV viruses. So this became a major breakthrough in quantifying the amount of antibodies present in the HIV patient. The next variant of this ELISA, which is called as the sandwich ELISA, is routinely used for checking the malarial antigens, or checking what kind of malaria the patient is suffering from and how much is the antigens which are present in its blood circulation. As we know that the malaria parasite is an intra-erythrocytic parasite, so it grows within the red blood cells. After the scission phase, during its life cycle, is burst out from the red blood cells, and with the bursting of this thing, it releases lots of malarial antigens into the blood circulation. So if these malarial antigens can be quantified, we can easily say that how much is the severity of the malarial cases are. So in this sandwich ELISA, antibody coated well was made, onto which the blood serum sample was loaded. As this blood serum sample contained the malarial antigen, they went and bound to these antibodies. Onto these antigen-antibody bound conjugate, the secondary antibody is added, which is conjugated with an enzyme, and this enzyme, what it does is it converts the colorless chromogenic substrate into a colored product. So we can easily measure the colored reactions over here, and with the help of the specialized spectrophotometer, we can measure the quantity of these colored reaction, and we can say that this much amount of these malarial antigens are present in the blood sample of this patient, and depending upon this, we can easily make out that the patient severity of the malaria cases. So over here. Now the next method which was developed based on this antigen-antibody interaction was called as the Western blotting. So Western blotting is a method which specifically identify the protein of our interest from a mixture of protein. So this method became one of the fundamental method to confirm the testing for the HIV patient. So this Western blotting method developed with the help of the antigen-antibody interaction. Now what exactly this Western blotting method is? It detects the specific protein. So from the protein sample which we have, we load these protein samples onto the well which is made up of agarose. Before loading onto the well, they are reacted with the SDS, which is sodium bodecyl sulfate, so they gets denatured, and there is a similar charge density which is imparted to all the proteins, and they start moving towards one of the sides of the electrodes. Now after the loading, the gel is switched on with the help of the electrodes which are present, and the electrophoresis start taking place. So based on their molecular weight, they get separated on the gel. Now from the gel, these separated proteins are then transferred onto the nitrocellulose membrane, and these nitrocellulose membrane are then incubated with the labeled antibody, which can be fluorescently labeled or radioactively labeled. So these radioactively labeled goes on to bind to specific portions of the nitrocellulose membrane where exactly the protein is present. So if we can see over here the autoradiograph in which there are specific black spots which are present, which shows that the specific protein of our interest to which the antibodies were specific is present in these particular sample. So Western blotting helped to detect the specific protein, and it is being actively used in most of the biotechnology and immunology based experimental assays. Now, the next major assay which was developed based on these antigen-antibody interactions were immunoprecipitation. So this immunoprecipitation is a method through which we can isolate a protein of our interest from the cell lysate. So suppose in a cell we want to isolate a particular protein or particular antigens from the whole cell. So what we can do is we first lyse the cell, then the cell lysate is transferred onto a tube. Onto this tube, the primary antibodies are added which are specific to that particular protein. To this primary antibody, a secondary antibody is added which has got a conjugated magnetic beads attached to it, and as these magnetic beads are attached, what happens is whenever we place a magnet into near to these tubes, what happens that these antigen antibody conjugate which contains the magnet beads comes to that particular side wall of the tube where the magnet is attached. Rest of the things can be eluted out, and the protein of our interest can then be eluted with the help of various techniques and various chemical reaction. So the protein of our interest with the help of the immunoprecipitation and the magnetic bead were isolated from these particular things. So this immunoprecipitation has also revolutionized the reaction processes or the sequences processes, and based on these particular immunoprecipitation reactions, various sequencing methods such as the chromato-immunoprecipitation and the sequencing process have started being developed. So the sequencing of the DNA or the RNA or these things to which the particular proteins or the binding factors are attached can be easily isolated with the help of the immunoprecipitation method. The next major method which was developed was called as the immunofluorescence, which revolutionized the field of immunobiology. This method was first developed by Albert Kunz in the year 1942. So this method was basically developed to track out the proteins which are present on the surface of the cells with the help of the labeled antibodies which are labeled with the help of a fluorescent tag. So these fluorescent tags can be fluorescein or rhodamine. These fluorescent tags, what the characteristic of these fluorescent tags are, they can absorb a light at a particular wavelength and they can emit out a light at a particular wavelength. So one of the fluorescent tag which is being used is called as a fluorescein. So what it can do is it can absorb the blue light and it can emit the yellow green light over here. So this fluorescently tagged radio labeled antibodies can be actively used to check whether the cell is having the protein of our interest on the surface or not. So there are two different methods which are being there for the immunofluorescence. One is called as the direct immunofluorescence, the other one is called as the indirect immunofluorescence. In this method, the cells are first immobilized on the microscopic slide, and then in the case of direct method, the fluorochrome labeled antibodies are then loaded onto these cells. So the cells which contains the antigens which are specific to these antibodies will bind to them, and as these antibodies contains fluorochromes, they will fluorescence light. So under the immunofluorescence microscopy, we can easily see the fluorescence or the fluorescence emitting cells over there. In the indirect method, first the cells are incubated with a primary antibody and subsequently the secondary antibody is loaded which contains a fluorescence. So this fluorescence also emits out the light. So this gives us the indirect method. Over here, the next third method which developed based on this particular principle was the immunohistochemistry. This method was basically developed to check or localize or visualize the distribution and the location of the specific cellular component within the cell or the tissue component without breaking the cell. In most of the assays, we were lysing or breaking, disrupting the cell, but in this immunohistochemistry, the cell has don't have to be laced up. We can easily check whether these particular antigens or the proteins are present on the surface of the cell or not. So in this case, suppose an antigen is present on the surface of the cell, we put the primary antibody, then we put the fluorescently labeled secondary antibody. As the fluorescently labeled antibody fluoresces out the thing, we can easily make out the thing. So over here, the colorectal cancer can be easily detected with the help of immunohistochemistry, and these immunohistochemistry as well as all the immunochemistry has revolutionized the immunobiology and the biotechnology field. So with this one, I end the lecture over here. Thank you very much.
Thank you, sir. Thank you so very much. You have given a deep insight into this very topic, and through you, we came to know how the blood groups, which we have learnt that the particular person has this blood group, but the RH factor, which plays a major role in deciding the positivity or the negativity of a blood group. With this note, thank you, sir. Thank you so very much, and hope in future also we would be carrying such lectures.
Thank you, ma'am.